So it's my great pleasure this evening to introduce this evening's keynote speaker, Professor Michael Levine. He's currently the Francis Williams Professor of Genetics over at UC Berkeley. He's also currently the head of the Division of Genetics, Genomics, and Development, and also the director of the Center of Integrative Genomics. Dr. Levine received his PhD from Yale University, where he studied steroid-regulated gene expression in Drosophila larvae. Then, as a Jane Coffin Childs Fellow, he did postdoctoral research with Walter Gehring at the University of Basel, where he co-discovered homeobox sequences in the homeotic genes, Antennapedia and ultrabithorax. After a second short postdoc with Jerry Rubin, he started his own lab at Columbia, then moved on to UCSD and to UC Berkeley. The Levine Lab has made a number of important discoveries over the last 30 years. They've studied gene regulation in the early Drosophila embryo, focusing studies on the SRIPE2 enhancer of the segmentation gene even skipped, and mechanisms underlying gradient thresholds of gene expression for the maternal morphogen Dorsal. Dr. Levine was awarded the Monsanto Prize in Molecular Biology from the National Academy of Sciences in 1996. He was elected as a member of the National Academy of Sciences in 1998. More recently, his lab has published pioneering studies on the use of paused RNA Pol II polymerase and on the existence of shadow enhancers in the fly genome. Since then, his lab has also done work in other organisms, including the sea squirt Cyona intestinalis, where they've been studying mechanisms of chordate development. So, Dr. Levine. There's only one click.
All right. I mean, if I didn't know any better, if I didn't know who Martha was talking about, I'd say, geez, that's kind of an impressive CV. But it's just me. Anyway, okay. So it hasn't been quite 30 years, adding more years. It's been about 27 and a half years that the lab has studied gene regulation in the early Drosophila embryo. And this first slide shows you why I just can't leave it. I feel like the Michael Corleone character in Godfather 3. Every time I try to get out, it keeps pulling me back. And this is the basis for being pulled back. It's just a beautiful system for visualizing gene expression. And here you're looking at the last two mitotic divisions in about a 2-hour Drosophila embryo, culminating with the formation of the cell cycle 14 after 13 rounds of nuclear division. At this stage, there's about 6,000 nuclei at the periphery of the egg. Cycle 14 runs for about an hour, and during this time, lots of different localized patterns of gene expression are established, which lay down the basic blueprint of the adult fly. And I'll mainly be talking about the cell cycle 14 1-hour window. You can see immediately one of the great advantages of the system for visualizing gene expression. It is a naturally synchronous system. All the nuclei are at the same phase of the cell cycle. And as I mentioned, the 6,000 nuclei present in the cell cycle 14 embryo are all arrayed as a monolayer at the periphery of the egg. So all the actions at the surface, and this really facilitates quantitative imaging. There's no need to focus into the murky interior of the embryo, the yolky regions, because all the good stuff is on the surface. And I think that'll become apparent in a few minutes. Many of our studies focus on the dorsal ventral patterning of the embryo. And this is controlled by a sequence-specific transcription factor called Dorsal. Dorsal is related to mammalian NF-kappa B. And here you're looking at a 2-hour side view of an embryo, staying with an antibody against the dorsal protein. And you can see it's distributed in a broad nuclear gradient with peak levels present in the nuclei present in ventral regions, the future belly of the fly, lower levels in more lateral and dorsal regions, the future back of the fly. And this dorsal nuclear gradient controls embryonic patterning through the regulation of approximately 50 target genes. And a few examples are shown on the next slide. This is a side view of an embryo that's just an hour older than the one I showed you on the previous slide. It was stained to show the expression of 4 different target genes that are directly regulated by the dorsal gradient. The gene visualized in yellow is activated by the highest levels of the dorsal gradient. Those visualized in blue, green, and red by by progressively lower levels of the gradient. So the dorsal gradient is getting right down to business to produce these different transcription thresholds of gene expression, and over the years we've learned quite a lot about how this process works. And the key are enhancers. For every localized stripe and band and cell-specific pattern of gene expression in the early fly embryo, you'll find that a developmental enhancer is responsible for producing these kinds of expression patterns. But that's not what I'm going to discuss tonight. Instead, I want to discuss mechanisms of robustness. How are these exquisitely precise patterns of gene expression deployed in large populations, natural populations of embryos, subject to changes in the environment, fluctuations in temperature, and to variations in genetic background? And I'm going to discuss two mechanisms of regulatory robustness: shadow enhancers and pause polymerase, and I'll begin with shadow enhancers. The next slide shows an example of a shadow enhancer, and the first evidence for this came from whole genome binding studies, initially ChIP-ChIP and then ChIP-seq assays, looking at the genome-wide distribution of the dorsal protein in early embryos. And in addition, we also looked at the distribution of two other sequence-specific transcription factors, Twist and Snail, that work together with dorsal to pattern the early embryo. And you can see, and I'm just showing you one gene, Sog, but of course we have this on a whole genome scale. And the Sog gene is activated by low levels of the dorsal gradient in lateral regions of the embryo. It's kept off in the ventral regions by the localized Snail repressor. I'm going to discuss this in a lot of detail in a few minutes. And you can see that the ChIP assays accurately identify the known intronic enhancer, which regulates the Sog expression pattern in response to the dorsal gradient. But to our surprise, there's also a cluster of binding sites identified that map over 20 kilobases upstream of the Sog transcription start site on the other side of this neighboring gene in a region where we had no reason to suspect that there would be meaningful regulatory information controlling Sog expression. But despite this relatively remote location, when this piece of DNA was tested for enhancer activity, it produces a pattern of expression very similar to that produced by the intronic enhancer and observed for the endogenous gene. So we have two different enhancers, not for two different patterns of expression, but in this case, two different enhancers for basically the same pattern of expression, separated by about 25 kilobases. And again, at the question of, why, what's the second copy, this remote shadow enhancer, doing? Mike Perry in the lab studied the regulation of the Snail gene. And I should say that it looks like on the order of 1/2 of all the critical patterning genes active in the early embryo, particularly during that critical cell cycle 14 stage that I mentioned, contain shadow enhancers. So it's quite a prevalent feature of developmental control genes in Drosophila. Now, Snail is activated by the highest levels of the dorsal gradient in ventral regions of the embryo. The Snail expression pattern defines the limits of the embryonic mesoderm. A little later in development, these cells will invaginate during gastrulation to form the internal mesodermal layer. The lateral limits of the Snail expression pattern define the boundary between the presumptive mesoderm and the lateral ectoderm, which will form the nervous system of the fly. And I'll discuss that in more detail in a few minutes. Snail is regulated by a proximal primary enhancer located just upstream of the Snail transcription start site. But surprisingly, the whole genome binding assays identified a shadow enhancer within the intron of this neighboring gene. We found the primary enhancer like 20 years ago. Never thought to look within this neighboring gene for additional regulatory information, but there's a shadow enhancer that was identified in the ChIP assays. The shadow enhancer maps 5 kilobases upstream of the primary enhancer. Despite the weird location, when attached to a LAGZ reporter gene, it produces an expression pattern very similar to that observed for the endogenous Snail gene. So just like in the situation I described for Sog, we have two separate enhancers producing more or less the same pattern of expression, separated by about 5 kilobases. And to get at the function, Mike Perry, grad student in the lab, looked at a series of back transgenes, about 25 kilobases, encompassing the Snail and neighboring TIM17 genes and all the associated regulatory sequences. And he mutagenized either the proximal enhancer or the distal shadow enhancer. And in some of the experiments, he replaced the small Snail transcription unit with a yellow reporter gene. Yellow has this nice big intronic sequence which permits quantitative identification of nascent transcripts. And an example of his analysis is shown here. So you're looking at the very early activation of the Snail back transgenes at optimal culturing conditions, room temperature 22 degrees. And you see that one enhancer is sufficient to give a normal pattern of gene activation. In this case, Mike has mutagenized the proximal primary enhancer, left the shadow enhancer intact, but you get the same result in the reciprocal situation, mutagenizing the shadow enhancer. And you see a normal pattern of activation for both the defective back containing just one enhancer and the normal back containing both enhancers. So one enhancer is enough under optimal conditions. But when you start looking at more stressful conditions, such as increases in temperature, one enhancer is not enough. And this is not an outlandish manipulation of the environment. I mean, flies in nature have to deal with 30 degrees all the time. They don't particularly like it, but normal populations deal with it just fine. And what you see is that this elevated temperature, the back transgene containing a single enhancer begins to break down. There's gaps in the expression, and the lateral limits start getting ragged. In contrast, the back transgene containing both enhancers continues to produce a normal pattern of activation. Looking at hundreds of embryos, this pattern of expression is not statistically different from the patterns observed at optimal conditions, at room temperature. So these types of experiments suggest that the shadow enhancer helps ensure that critical patterning genes like Snail are turned on in the right place, in the right time, in embryo after embryo, in large populations of natural embryos subject to all sorts of variation in the environment and genetic variation. Natural populations have lots of genetic variation as well, and not shown here is that two enhancers help buffer against genetic background changes, like removal of one copy of Dorsal. No problem if you have two enhancers, but a breakdown when you have just one. Now, we haven't been able to prove this idea, but the thinking is that the shadow enhancer arises from some kind of cryptic duplication event, cryptic duplication of the primary enhancer, kind of like a gene duplication. And that second copy gets fixed in natural populations because it gives immediate value. It gives robustness in response to variation. Once you have that second enhancer, we sometimes find that the two enhancers could work synergistically to give more precision in the core patterning process. And I'll show you an example of that, where having two enhancers actually makes, in any given embryo, not in a population, but in every given embryo, the patterning process a little more precise. And we see that for the so-called gap genes, targets of the Bicoid gradient. So shadow enhancers aren't just seen for dorsal ventral patterning, they're also seen for genes important for segmentation. And these are controlled by the famous Bicoid gradient, with peak levels present at the anterior pole, lower levels in more posterior regions. This Bicoid gradient controls segmentation in part by regulating a series of gap genes. And all the gap genes have shadow enhancers. In fact, just about every gene that's responding primarily to a regulatory gradient contains shadow enhancers. Some examples are shown here. These are 3 of the 5 or 6 gap genes: Hunchback, Krupel, and Knurps. They all have shadow enhancers, and I want to just focus on the regulation of Hunchback expression. So Hunchback shows the simple on-off switch in transcription. Both high and intermediate levels of the Bicoid gradient emanating from the anterior pole are sufficient to switch Hunchback on. Low levels of the Bicoid gradient, more posterior regions, are unable to switch on Hunchback. And this on-off switch depends on a proximal enhancer straddling the P2 promoter, which was identified over 20 years ago in the labs of Nusslein Volhart and Gary Struhl, I believe, collaborating with his older brother back in '89. And this proximal classical enhancer contains several high-affinity Bicoid binding sites that are evenly spaced. And it's sufficient to produce more or less a normal on-off switch in Hunchback expression. Mike Perry in the lab, looking through whole genome data, identified a shadow enhancer located 5 kilobases upstream of the classical enhancer, which has evaded detection all these years, but it produces a pattern of expression very similar to that produced by the proximal classical enhancer. So just like in the case of Snail, we have 2 enhancers, a proximal and distal enhancer, that produce very similar patterns of expression separated by about 5 kilobases. And what Mike found is that the 2 enhancers can work synergistically to give very sharp borders of gene expression. And an example of his analysis is shown here. So these are a series of back transgenes looking very early at the activation of hunchback expression in response to the Bicoid gradient. These activation profiles are observed within 20 minutes of the formation of the Bicoid gradient. Very rapid response. And here you see back transgenes containing just one enhancer or the other. Here the shadow enhancer is mutagenized, leaving the classical enhancer intact. Here's the reciprocal situation. The classical enhancer was mutagenized, shadow enhancer intact, and then both enhancers left alone. And you can see that the back transgenes with single enhancers give initially kind of erratic, heterogeneous patterns of activation with fairly indistinct borders. But the two enhancers together give a very nice homogeneous pattern of transcriptional activation with a fairly sharp border. So shadow enhancers, I think, are used both to confer robustness in natural populations. They can then be incorporated back into the core patterning machinery to make it a little sharper and a little more precise. I mean, it's a bit of a chicken and the egg question. Do you need the shadow enhancer to make patterning better? I believe the shadow enhancer comes along to give you robustness in natural populations. That happens first, and then it's incorporated into the patterning machinery. But that's a difficult thing to test. In addition to shadow enhancers, the second mechanism of regulatory robustness I want to discuss is paused RNA polymerase. And for those of you who have heard me talk about this before, Kevin, I know, is nervous when I talk about paused polymerase. You're going to see that I've backpedaled to a very moderate position regarding exactly what paused polymerase is doing in development. So the first evidence for paused polymerase came from whole genome Pol II ChIP-seq assays and ChIP-chip assays done in collaboration with Julia Zeitlinger while she was a postdoc in Rick Young's lab at MIT. And here you see two genes, tail-up and pan-A, prior to their activation in the early embryo. You see that tail-up has a little peak of polymerase bound to the promoter region, but pan-A does not. There are two pan-A promoters, a distal and proximal promoter. Neither of these have detectable association of polymerase prior to the activation of the gene in development. And I should also say that we see this association of polymerase, for example, on the tail-up promoter, in tissues where the gene is not turned on. So tail-up is normally turned on in the dorsal ectoderm, but we see this paused polymerase in the mesoderm where the gene is never turned on. Okay, fine. Most of the critical patterning genes active in the early fly embryo have paused polymerase. Pan-A is somewhat exceptional. So what does the paused polymerase do? It fosters rapid and synchronous patterns of gene activation, and I'll show you some evidence for that. So here you see the mature patterns of tail-up and pan-A expression. Tail-up is the Drosophila equivalent islet 1. Pan-A is the fly's GATA4. And these two target genes respond to a BMP signaling gradient emanating from the dorsal midline, the dorsalmost regions of the embryo. You're looking right down on the back of this late cell cycle 14 embryo. And these two genes are needed for the subsequent patterning of the heart later in development. Tail-up is activated only by high levels of the BMP signaling gradient, but pan-A by lower levels. And so pan-A shows broader lateral limits of gene expression. There's different repressors impinging on the anterior-posterior pattern, but the main point here is that these different dorsal ventral limits of expression depend on differential responses of these genes to a BMP signaling gradient emanating from the dorsal midline. Now, we've isolated the enhancers underlying the tail-up and pan-A expression patterns. We know a fair amount about how you get these different spatial patterns. What is far less well understood in general in development is the timing of gene expression. How do you control temporal patterns of gene expression? And that's what I want to talk about. Now, here you're looking at the very early activation of tail-up and pan-A shortly after the gene is first switched on, several minutes after the first detection of any nascent transcripts. And you see the tail-up comes on in a much faster and more synchronous fashion than does pan-A. So over 80% of the nuclei, which will eventually show expression of tail-up, show nascent transcripts within several minutes of activation. In contrast, just several percent of the nuclei which will eventually show expression have detectable nascent transcripts within the first few minutes of activation. And so the tail-up gene with the paused polymerase comes on more synchronously and more rapidly than does PANA, which lacks the pause polymerase. And this can be seen more quantitatively in these activation profiles. And these experiments are very, I mean, I know a lot of you guys do a lot of work, but this is an unusual amount of work. Poor Jacques Bothma, the graduate student in the lab. I don't know if you guys saw the Woody Allen movie Sleeper, but for a while Woody Allen's kind of hiding in the orgasmatron. That's how poor Jacques looks after he's been in the confocal room working on these activation profiles. He kind of comes out like that. Anyway, so at least he tells me it's hard. So what you're looking at here are the fraction of, on this axis, you're looking at the fraction of nuclei, embryo by embryo, that show nascent transcripts for tail up in blue and PANA in black. And we consider the 80% point, 80% of the nuclei showing expression is when the gene is basically functionally switched on. And you see that PANA comes on very abruptly, very synchronously, very rapidly. It comes on 20 minutes ahead of PANA, which takes its sweet time before reaching this 80% point. Now, 20 minutes may not sound like a lot, but remember, this whole thing is 1 hour. All the action in cell cycle 14 that's basically patterning the embryo occurs over a course of 1 hour. So this is a significant temporal difference. So tail-up comes on 20 minutes faster than PANA, and again, we're defining activation as when you achieve 80% of the nuclei that will eventually express the gene showing nascent transcripts. And this difference, this temporal difference, is really a property, and this surprised me, is a property of the promoter, not the enhancer. I mean, I've never seen an enhancer that I haven't liked. You know, I like enhancers. I've done enhancers for 25 years. But it looks like enhancers may be more important for the spatial control of gene expression, at least in this system, whereas the promoter is really important for the temporal patterns of expression, for different timing in gene activation. And some evidence for that is shown here. So this is a back transgene for the tail-up locus, about 60 kilobases, includes the tail-up transcription unit and associated regulatory sequences. And its pattern of activation is pretty similar to that observed for the endogenous gene. Any difference here is that we're looking at heterozygotes in this experiment, and here we're looking at both copies, both alleles, so it looks a little faster. But it's basically the same dynamics. And here you're looking at the activation profile of a modified tail-up back transgene that was manipulated by a postdoc in the lab, Munia Lagha, and she basically replaced the tail-up core promoter region, about 100 nucleotides, centered around the +1 transcription start site. She replaced it with the PANA promoter. And now she observes the PANA, not the tail-up, but the PANA dynamics. It is slow and stochastic and 20 minutes delayed relative to the onset of tail-up expression. And so again, this activation profile is of a tail-up back transgene having all the tail-up regulatory sequences, all the proper enhancers, but just a 100 nucleotide region of the promoter has been exchanged for PANA. So it looks like the promoter is really sufficient to dictate these different temporal profiles of activation. But it's not just as simple as pause genes come on fast, non-pause genes come on slow. It looks like there's really a spectrum of pausing. And so this shows normalized Pol II read counts from whole genome Pol II ChIP-seq assays for a number of different genes. And tail-up, I've mentioned, is paused. PANA is not. This dashed line corresponds to where we had previously identified pause versus non-pause genes based on the so-called stalling index, the ratio of Pol II in the promoter region versus the main body of the transcription unit. And so tail-up, PANA, and SOG were seen to be paused, but you can see that they're not equivalent. And again, these are normalized reads, I think reflecting the stability of pause polymerase at these different promoters in early embryonic tissues where the genes are not turned on. Okay, so it's not complicated by active transcription. We're looking at silent tissues. And you can see that tail-up has 2 times more Pol II read counts than the snail and 3 times more than SOG. Similarly, not all non-pause genes are equivalent. There are more Pol II read counts at the non-pause Thisbe gene as compared to PANA. This spectrum of Pol II association at these different core promoters is reflected by the spectrum of activation in the following experiment. Munia took the same pan-A enhancer, which directs activation throughout the dorsal ectoderm of the early embryo, and placed this upstream of the minimal promoter regions from these 5 genes and several others, driving a yellow reporter gene all inserted into the same region of the genome, so permitting direct comparisons. And you see a very nice correlation between the relative Pol II binding scores and the timing of activation, with pan-A being the slowest to get turned on, tail-up being the most rapid, Snail showing an intermediate level of activation, and Thisbe, like pan-A, we call non-pause, but clearly coming on more rapidly as compared with pan-A. And the activation curves are shown here. So this is across the 1-hour time window of cell cycle 14, and you can see with the same pan-A enhancer, just changing the 100 base pair minimal promoter sequences, we're getting very different temporal profiles of gene activation across cell cycle 14. So we conclude from this, at least regarding DPP, BMP signaling readouts, that the enhancer is important for the spatial limits of gene expression, whether the gene is activated by high or low levels of BMP signaling, but the promoter region is critical for the timing of activation. And this is summarized on the last slide of this series. And this just kind of reminds me to point out the emerging evidence, which I find very interesting, coming from a number of labs, including Eileen Furlong's lab, so she may talk about this tomorrow morning, that genes don't just suddenly get turned on in development, but they are prepared for activation. So for example, the enhancer in the early fly embryo is often prepared for activation through binding of a ubiquitous protein called Zelda. That's not enough to turn the gene on, but it's thought that Zelda works as a sort of pioneer factor to open up the enhancer so that it will be smoothly induced when the right signals become available. And similarly, we think of pause polymerase as priming the promoter in the same way that pioneer factors like Zelda prime the enhancer. It's just getting the gene ready for smooth and rapid induction. And I've showed you evidence, depending on how much Pol II you have at the promoter, whether it's stably paused or there is no pause polymerase, determines the timing of activation, with promoters containing sort of an unstable form of pause polymerase, giving an intermediate activation dynamic of expression. So again, it looks like the promoter is important for the timing of gene expression. Now it turns out, so I've discussed shadow enhancers and pause polymerase, and it turns out, like, if I write a list of what I consider to be like the top 10 movers and shakers of embryonic patterning. I'm not going to divulge that list because I don't think any two fly people are going to agree on that. But my list, my secret list of the top 10 genes driving early embryonic patterning, most of those genes, 7 of the 10, have both strongly paused polymerase and shadow enhancers. And I want to discuss one example. And that's the Snail gene, which I briefly mentioned before. So here's Snail. As I mentioned, it's activated by high levels of the dorsal gradient in the ventral mesoderm and has sharp lateral limits. That sharp border of Snail expression delineates the boundary between the future mesoderm and the lateral ectoderm, which will form the nervous system of the fly. Snail has paused polymerase, fairly strongly paused, not the gold standard like tail-up, but the second most stably paused polymerase, very rapid pattern of activation when that promoter was tested in isolation. And as you recall, it is regulated by both a proximal primary enhancer and a distal shadow enhancer. This combination of pause polymerase and a shadow enhancer ensures very rapid induction of gene expression. And I'm going to tell you why rapid induction is important and then show you the evidence. This sharp Snail border depends on mutual repression between Snail and several different neurogenic repressors restricted to lateral regions of the embryo. Snail encodes a zinc finger repressor, which directly represses these neurogenic repressors. I mean, we know one of them is VND. There's several of them, and we haven't identified them all. So the idea is that Snail represses the neurogenic repressors, and these in turn feed back and repress Snail to form the sharp order. Simple bistable threshold switch dependent on mutual repression. You see this over and over again in the early fly embryo. But this bistability, I believe, comes at a cost. And the cost is you got to turn on Snail really fast. Because if Snail is slow out of the gate, if the Snail repressor function is impaired, we know that these neurogenic repressors will take over. They will wipe out that border. They will become expressed in these ventral regions and literally convert mesoderm into neurogenic ectoderm. So bistability and mutual repression is fine for a sharp border, but you got to deal with the problem of really, rocking and rolling, getting that gene expressed in a hurry in the early embryo. And I think that's what the combination of the strongly paused polymerase and the shadow enhancer does for you. Now, to get at the quantitation of this bistability, a grad student in the lab, Alistair Boddicker, has done quantitative in situ hybridizations. And without going into the kind of dull details, he took a fluorescent probe, hybridized. This time we're not looking at nascent transcripts, we're not looking at de novo transcripts, we're looking at stable Snail cytosolic mRNAs. He did a variety of high-resolution confocal imaging and various computational deconvolution methods to look at individual hybridization dots. And we have pretty good reason to believe that each of these hybridization dots corresponds to a single mRNA. So all that rigmarole, high resolution blah, 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 and deconvolution this and that, and you put it in the computer, and well, you count the dots. Okay. So that's all I get. I get that part. So here you see a high magnification view of a massaged image, 4 consecutive nuclei. These 2 are in ventral regions in the mesoderm. Did I sound a little like Potashny there? That was kind of a Potashny imitation. And wait, here, let me do it properly though. Blah blah blah. Gizmos. Anyway. And so here Snail is on. Here is just on the other side of the border. Snail is off. And you can see the red dots. There's more on the apical side of the nucleus than the basal side, and this is true for a lot of mRNAs encoded by critical patterning genes. They're asymmetrically localized within the cell. This was first noted by David Ish Horowitz over 20 years ago, and we still don't know what it means. To this date of my knowledge, nobody's done the experiment of delocalizing these RNAs so that they're expressed uniformly throughout the cytosol and seeing what that does in embryonic patterning. But they do tend to be localized. And so the next slide shows the numbers. So this is counting. Each dot is the number of Snail mRNAs, or hybridization dots, within a nucleus of a given embryo at this indicated stage. And we're looking at nuclear cleavage cycle 13 and nuclear cleavage cycle, cell cycle 14. At the onset of cycle 13, there's an average of 50 Snail mRNAs per cell, and this rises to an average of 120 mRNAs at the conclusion of cell cycle 13, which has an interphase of only 15 minutes. Then the nuclei divide, and of course this now comes down to an average of 60 Snail mRNAs per cell at the onset of cell cycle 14, and this steadily rises over the first 25 minutes, about the first half of cell cycle 14, to an average of 180 Snail mRNAs per cell, and that's steady state. That's the steady state, and this will now persist throughout the remainder of cell cycle 14 and into gastrulation. And so with this information, we can calculate synthesis rates, and Snail is made really fast. Okay. So as I mentioned, 180, steady state of 180 mRNAs per cell by the midpoint of nuclear cleavage cycle 14. We estimate that over 20 mRNAs are made per minute, assuming a half-life of 8 to 10 minutes. That's the trickiest calculation. We think that the half-life is more like 7 to 8 minutes. And I can tell you how we arrive at that number if you're curious at the end. And so with those numbers, we estimate one Snail mRNA is made every 5 to 8 seconds per allele. And that's within a factor of 2 of the theoretical limit. That's really fast. The theoretical limit in Drosophila is constrained by a Pol II elongation rate of 1 kb per minute. And this was calculated by John List many years ago and confirmed in a variety of studies. I'll show you some evidence for that in a minute. And so basically, because of the large Stokes radius of the polymerase complex, you can only load polymerase every 80 nucleotides. So that's one polymerase every 4 seconds. And so even if you get bumper-to-bumper traffic, maximum density, a polymerase every 80 nucleotides, all moving at the maximum rate of 1 kb per minute. That's an mRNA every 4 seconds. And we're getting an mRNA every 5 to 8 seconds. So that's really very fast. And I think that's to deal with that bistability problem, the mutual repression I talked about, and it's because Snail has a combination of the pause polymerase and the shadow enhancer. So altogether, I would say that the pause polymerase and the shadow enhancer is a potent 1-2 punch that controls the robustness and precision of gene expression in both time and space. Now, Madam Chairman, how many more minutes do I have? 5. Oh my God, I'm talking so slow. Okay. I'll pick up the pace. All right. I want to end on my favorite topic, and that's transcriptional repression. Now, I just talked about Snail. It's very rapid synthesis in the ventral regions of the embryo. Snail represses a lot of genes, including this one here called Sog, which I've mentioned before. Sog is activated by low levels of the dorsal gradient in lateral regions of the embryo. And Snail represses about 50 target genes, including Sog. Now, when Sog first comes on, it's activated very early, before this critical threshold levels of the Snail repressor. So you see Sog expression in both lateral and ventral regions. Then Snail kicks in and clears Sog expression out of the ventral mesoderm. And Jacques has done high-resolution imaging of this progressive repression of Sog out of the ventral mesoderm. And the basic rationale of the experiment is shown here. Sog is a fairly large transcription unit, 20 kilobases in length, dominated by 2 large introns, a 5' intron and a 3' intron. And Jacques made a green 5' intronic probe and a red 3' intronic probe, mixed these together, hybridized into embryos at different stages to visualize Sog expression and repression. And so basically what you expect to see is that when the Sog gene first comes on, you only get hybridization with the green probe because you only have 5' nascent transcripts. But after a delay, the polymerase enters the 3' intron, it splices out the 5' intron, finally reaches deep enough into the 3' intron to cross-hybridize with the 3' intronic probe. And so you get nascent transcripts that are stained red. The nuclei are yellow because you have nascent transcripts with a mixture of either 5' or 3' intronic sequences. Now the Snail repressor kicks in after you get the stable activation of Sog, and there are 2 possibilities. One is that Snail blocks Pol II elongation, so the polymerases fall right off the template as soon as Snail gets to the Sog DNA template, and you go from yellow to no signal. The gene is just immediately silenced. That seemed unlikely. The more likely scenario is that the Snail repressor works at the promoter. It blocks the activation of polymerase, either the recruitment of polymerase or the release of pause polymerase from the promoter. And this means, according to repression by initiation, that the elongating polymerases that were released from the promoter prior to the onset of Snail repression will complete transcription before the gene is fully turned off. And this should be seen as a red flash. So if you wait long enough, during the repression by Snail, you should see an intermediate phase of red-only hybridization signals. These are the final polymerases completing transcription of the Sog gene that had been released minutes earlier after the onset, prior to the onset of Snail repression. And this is what we see. So here's the actual data. You're looking at progressively older cell cycle 14 embryos, early to late. When the gene first comes on, you see only green nascent transcripts because the polymerase is only transcribed through the 5' intron. The gene comes on fairly synchronously, fairly early, as I've already shown you. Then after a 10-minute delay, the nuclei turn yellow. That's because the polymerase has now moved into the 3' intron. You have a mixture of nascent transcripts, red and green, 5' and 3' intronic sequences. And this delay of 10 minutes is consistent with the Pol II elongation rate of 1 kb per minute, as I mentioned. Now the Snail repressor kicks in, and you see this red flash. You see this intermediate stage where you get red-only hybridization signals. Those are the final elongating polymerases completing transcription of the Sog gene that had been released prior to the onset of Snail repression. This is strong evidence that Snail doesn't work by blocking Pol II elongation, but blocks the initiation, the activation of polymerase at the promoter. Because of this mechanism, because the elongating polymerases can complete transcription, there is a repression lag. It takes 20 minutes between the onset of Snail repression and the inhibition of Sog expression in the ventral mesoderm. And the length of the repression lag is commensurate with the size of the gene. Bigger the gene, the longer it takes for repression. And it's possible that a major function of microRNAs is to get rid of the unwanted mRNAs that are produced after the onset of repression. We calculate that during the 20-minute period of repression lag, between 50 to 100 unwanted Sog mRNAs are synthesized in the ventral mesoderm. And this could cause a problem in the dynamics of embryogenesis, where things are moving fast, and it's possible, if you got to get rid of it, if that's going to cause a problem, you need a post-transcriptional mechanism, such as a microRNA. And I note that among the longest genes in the Drosophila genome, UBX and Antp, which are on the order of 100 kilobases in length, and take an hour and a half to repress through this mechanism, are notoriously regulated by multiple microRNAs. In the last minute? Because I know it's stressful to be the chair, and you got an old guy who won't get off, and what do? Do you get the hook out? Do you open the trap door? As long as you give me a shot of bourbon, however you want to dispose of me is fine. Okay. I can't get enough of those Struhl brothers. Let's face it. So I just want to end. I want to end with Polycomb. And this is what this is the only reason why Kingston came to the goddamn talk. It's because I said I was going to talk about Polycomb. Okay. So in 2 minutes. After Snail, finally, after the repression lag, inhibits Sog expression of the ventral mesoderm, pause polymerase is reestablished in the mesoderm. Okay, so at 3 hours after fertilization, Sog is off in the mesoderm, but we know in that tissue that there's pause polymerase at hour 3. Yet, Sog is never going to come on in that tissue again. Okay, and at about 7 to 8 hours after fertilization, 4 to 5 hours after the transient Snail repressor has finally inhibited Sog in the ventral mesoderm, we see a loss of the pause polymerase from the Sog promoter in the mesoderm, which makes sense. Gene's never going to come on. Throughout the life cycle of the fly, after hour 3, why have the pause polymerase there? We believe that's priming the gene for activation. You want to get rid of it. Converting a repressed gene with pause polymerase into a repressed gene that is silenced, that lacks pause polymerase, I think, is a major job of the Polycomb repressors. And I'll just show you some evidence for that and get off. All right, so here you're looking at so-called cuticle preps. Basically, this is the ventral epidermis or skin of advanced staged embryos, wild type, and a Polycomb mutant studied by Gary Struhl, a shocking 30 years ago. And I should point out that Gary is Kevin's younger brother. 30 years ago. I realize he did this in the womb, but nonetheless, it's a long time ago. And you see for ESC the typical Polycomb mutant phenotype. All the anterior body segments, the anterior abdomen, thorax, and head segments are transformed into a repeating series of eighth abdominal segments, of A8 segments. And this is due in part to the de-repression of the posteriormost Hox gene called abdominal B. Abdominal B is normally transcribed at the highest levels in these posteriormost regions that form A8, but in Polycomb mutants such as ESC, abdominal B is transcribed at high levels throughout the embryo, and this transforms the anterior segments into an A8 identity. ABDB works as a transcriptional repressor. It shuts off anterior Hox genes such as Antp, which is normally transcribed in the thorax. In Polycomb mutants, ABDB is expressed everywhere. It shuts off Antp, and that helps the conversion of thoracic segments into extra copies of A8. And so a postdoc in the lab, Vivek Chopra, did whole genome Pol II ChIP-seq assays and collaborated with Leighton Core in John Liss's lab to do whole genome growth-seq assays to look at the distribution of Pol II and ESC mutants. And the reason why we used ESC, it's the only Polycomb mutant where we can do these experiments. It shows maternal inheritance, and this just means we can get large quantities of homogeneous mutant tissue to do the whole genome assays. So this just shows, and this is like just 2 more slides. So this shows the binding of polymerase to the proximal Antp promoter. In wild type, you see a little bit of pause polymerase, but to our surprise, in the ESC mutants, you see an increase in the amount of pause polymerase. Remember, in ESC mutants, this anterior Hox gene is repressed by ABDB. It is off in the ESC mutants, so we thought there'd be no pause polymerase, but instead, there's more pause polymerase in this mutant, despite the inhibition of Antp expression. And this trend of more pause polymerase in the ESC mutant is observed for over 1,400 genes, for about 10% of the genes in the Drosophila genome. And this is a compilation of whole genome grow-seq assays, nuclear run-on assays, and this really grieves me. This is a problem with the cultures between molecular biology and computational biologists. The transcription start site is denoted as 0. There's no 0 in transcription. That is the +1 transcription start site, and I apologize for not changing that. And I got to tell you, I got a little OCD, and it bothers me to see it. I can't even look at it. That 0 really upsets me. But in any event, the black bar shows the pause polymerase on average over all these genes at the promoter of wild-type embryos. In red, you see the ESC mutants. This is the plus strand for genes transcribed that way. This is the minus strand for genes transcribed that way. You're looking at -1 kb and +1 kb, a 2 kb window. And you can see a very clear-cut trend of increased pause polymerase in the ESC mutants. This correlates with the loss of a position nucleosome at the core promoter, and this is based on whole genome micrococcal nuclease assays, again, at these 1,400 genes, and there's that hideous 0 once again. In wild type, you see a pretty nice position nucleosome within the first 100 nucleotides of the transcription start site, but in ESC mutants, this is basically lost. And we think that's the big event for the Polycomb repressors, is that they're positioning a nucleosome at the core promoter, and this kicks out the pause polymerase. In ESC mutants, you lose this position nucleosome, and now you get an increase in pause polymerase. And the last data slide just summarizes this. So I want you to think about the Sog gene right after the transient Snail repressor has shut it off in the ventral mesoderm. Initially, the pause polymerase is restored on the Sog promoter in the mesoderm, although the gene's never going to come on in that tissue again. And then several hours later, this pause polymerase is replaced by a position nucleosome at the promoter. And we think that is not the only thing, but a major function of the PRC2 repression complex. And of course, you're going to hear a lot more about this tomorrow night from Bob Kingston. And so what we think is going on is that when you take away this critical component of the PRC2 complex, the ESC mutants, you now lose this position nucleosome and get enhanced pause polymerase at the promoter. So I'm going to end by thanking the people who did the work. I discussed 3 topics: shadow enhancers, pause polymerase, transcriptional repression. The work on shadow enhancers, the evidence that they confer robustness in response to fluctuations in temperature and genetic background, that they produce more precise borders of gene expression, was done by a graduate student named Mike Perry. Evidence that pause polymerase, there's a spectrum of pausing, and that this produces a temporal sequence of gene activation, that the promoter is important for timing of gene expression during development. This was done by a postdoc in the lab, Munia Laga. The bistable switch and the mutual repression of Snail, the formation of the sharp border, that quantitative analysis was done by Alistair Boddicker, a graduate student in the lab. The repression lag, that Snail does not interfere with elongating polymerases but blocks Pol II activation. This was done by another graduate student, Jacques Bothma. Remember, he was the one in Woody Allen's Orgasmatron. And then finally, the bit about Polycomb silencing, that the PRC2 complex might convert repressed but paused genes into repressed but silent genes lacking pause polymerase due to the positioning of a nucleosome. That was done by a postdoc in the lab, Vivek Chopra. And then these are our collaborators, mainly on the pause polymerase work: Julia Zeitlinger at the Stowers Institute, Leighton Core, postdoc in John Liss's lab at Cornell, and David Wilmore at Penn State, and I apologize for going over. Thanks for your attention.
So we have time for a number of questions.
Hi. Does the efficiency of the shadow enhancer for Snail, is it dependent on whether its host gene that it lands in is transcribed or not?
Yeah, I mean, so these are doing like the back transgenes and inserting that into different positions, and you're wondering about the influence of the neighboring chromatin depending on where the transgene inserts in the genome?
So I thought, so the shadow enhancer for Snail, it's inside another gene, isn't it? So if that gene that it's inside, if it's transcriptionally active, does it affect?
Gotcha. So right. So the Snail, specifically the Snail shadow, and by the way, this is not an unusual location for shadow enhancers. About half of them that we found are within neighboring genes. You know, within the introns of neighboring genes. You know, it's not a place I would have looked, but they are often seen there. In the specific case of Snail, that neighboring locus, TIM17, is inactive in the early embryo and isn't turned on till later in development. By the time it's turned on, that shadow enhancer has run out of juice. The activators are no longer present. It no longer regulates Snail expression. So in that particular case, there doesn't seem to be a problem with interference between the host gene and the shadow enhancer. But I can't say that that's true for all of them, but in that specific case.
So regarding the last story with Polycomb.
Yeah.
So do we have evidence for the appearance of a nucleosome there is really the cause of the falling of the polymerase?
Causality in life and in chromatin. I mean, geez. Now, why are you holding me accountable for causality when there's a whole industry of people doing it and they don't ever worry about causality? I don't know. Look, I wish I could say that the primal effect of removing the PRC2 complex or compromising the function of the PRC2 complex is the loss of the position nucleosome at the promoter. But we don't really know that. I mean, it is coincident. If we look over time in individual tissues at nucleosome occupancy at these pause promoters versus polymerase occupancy, either by Pol II ChIP-seq assays or grow-seq assays, it's more or less coincident and reciprocal. Seems like it's occurring about the same time during embryogenesis. So I can't tell you which is causal. And certainly, Karen Adelman proposed in a very lovely Cell paper of 2010, she showed that a number of developmental control genes in Drosophila like to have either pause polymerase or a position nucleosome at their promoters. And they are competitive. It's mutually exclusive. And so when you have one, you don't have the other. So, what I'm arguing is that somehow the PRC2 complex is influencing that balance between pause polymerase and the nucleosome at the promoter, favoring the positioning of the nucleosome somehow. And I don't know if this is going to square with, Bob's data on, the role of PRC1 and PRC2 in compacting chromatin or not.
Mike?
We'll find out. I would like to think yes, but we'll have to wait till tomorrow night. Bob says yes, but come back tomorrow night.
But holding on that theme, Mike, over here. On the same theme, one, in the direction...
The causality theme?
Yeah. You have a series of genes that you showed us at the beginning of the talk that some are rapidly inducible and have polymerase bound there and some do not. Is the basis of those genes, do the ones that do not get induced, do they have position nucleosomes over their promoter and do the others not? And can you manipulate the nucleosomes, and does this lead to a change in polytoxins?
That's a great question, and now I know who to exclude as reviewer number 3 from the forthcoming submission. In fact, micrococcal nuclease assays, I'm not sure we're looking at exactly the same thing in the early embryo and the later stages. Let me emphasize that this flip between pause polymerase and the position nucleosome, however that works mechanistically, involving PRC2, that's occurring fairly late in development, between 7 and 10 hours after fertilization. The spectrum of pausing I showed for genes such as tail-up and Panier, that's occurring in the 2 to 3 hour embryo. And we haven't yet collated all those data sets. I think it's going to be the same phenomenon, but we haven't shown that. But to answer the specific question, do the genes that have less pause polymerase, that tend to come on slower and later in the early embryo, do they tend to have more position nucleosomes at the promoters compared to pause genes? The answer seems to be yes, based on global micrococcal nuclease assays and different dorsal ventral patterning mutants in the early embryo.
So, Mike,
yes.
After this response to the causality, I'm a little scared to ask a question using the M word, mechanism.
Mechanism.
So,
yeah.
And so I'm going back I
thought by M you meant mystery. Anyway.
Whatever you want to call
or
it.
magic.
But I want to go back to the shadow enhancer. So it seems to be paradoxical that when you have the temperature effect, if you miss one of the two enhancers, you start to get holes in your pattern.
Yeah.
Whereas in the patterning anterior-posteriorly, when you have only one enhancer, now you start to see dots appearing where they're not supposed to be. So that's sort of the opposite thing.
Ectopic expression.
And so what's going on there?
Yeah, I'm not sure it's going to be a consistent difference between anterior-posterior and dorsal ventral patterning, but we do believe that the shadow enhancer is not absolutely identical to the classical enhancer with respect to what repressors bind to. With respect to the activators, it's Bicoid, it's the same Bicoid logic, same kind of binding sites. In fact, if you look at the core sequences of those Bicoid binding sites, the ones in the classical enhancer look most like the ones in the shadow enhancer than any other Bicoid-dependent enhancer in the genome. So these enhancers are definitely somehow related, but it does look like in that one particular case that the shadow enhancer has some repressors that we haven't been able to identify that ensures that you don't get ectopic activation in more posterior regions. That's what you're responding to. And I don't know if that's just a quirk of that particular gene. It doesn't look like a trend. Most of the time, what we see when we take away one or the other enhancer or start looking at temperature differences are just heterogeneities in the normal domain of activation. And that hunchback is a little exceptional in giving the ectopic expression. I should say in the case of that hunchback story, as far as removing one enhancer, it's just like what I said for Snail if you start going to high temperature. I was showing all that at normal temperature. So at any temperature, 2 enhancers are better than 1 in giving precision of the pattern. I'm afraid to let Kevin answer the question. Yes, good.
Hi.
Yes. Well, I don't know if Eileen's any better. The tough ones are coming late.
Yeah, so I just wanted to get back to the timing and the release of pausing. So, I would have thought that there would be a transcription factor, some subset of transcription factors may be involved in release of pausing. But, I think the promoter swap experiment is just so, that was just such a surprising result in some way that really suggests there's some motif in a subset of promoters. So, do you know, what is it at the promoters?
Yeah, we haven't progressed much beyond where we were a few years ago in terms of doing the bioinformatics analysis of pause versus non-pause genes. Now, and that revealed.
Zelda.
Yeah, and I can tell you, paused promoters tend to have closely associated GAGA elements, which are known to kind of open, recruit. The GAGA-associated factor, GAF, or trithorax-like, which in turn recruits NERF, a SWI-STIF remodeling complex that displaces nucleosomes. They tend to have good initiator elements. They tend to lack tautoboxes. They tend to have a GC-rich version of the Katanaga downstream promoter element, or DPE. And those features are kind of predictive. You can go in and you can find other pause promoters, bioinformatically in silico using those features. But I don't think the story is so simple in light of, obviously, your recent work, Julia Zeitlinger's work, which is, you kind of prepare a gene an hour before it's going to get turned on. So a gene that we would say is not paused in the 2-hour embryo because it's turned on in a late muscle cell will be paused 2 hours later, 1 hour before it's turned on. And so I'm not sure we did the bioinformatic analysis right in just simply saying these are paused and those are not paused. I think it's got to be done on a stage-by-stage basis, which we haven't yet, done. And as far as, and since this is related to the topic that I promised I'd mentioned to Kevin, my backpedaling is, I used to think that the pause polymerase was everything and that the regulation was all about release of the pause polymerase. And the, proteins like PTEF-B and the so-called super-elongation complex of proteins that phosphorylate serine 2 on the pause polymerase, which releases it from the promoter, that at every round of transcription, the polymerase, arrests at the pause site, and it's the elongation factors at every single round which is responsible for gene activation. I'm beginning to think, and we don't have definitive evidence on this, but I'm beginning to think that just as Karen Adelman has argued, that the pause polymerase is there to kind of keep nucleosomes off and to keep the promoter in a sort of an open state. And that after that, it's just classical recruitment the way people have always thought about the problem. Because, one pioneer transcript made from that pause polymerase doesn't buy you much. You need like 50 rounds before you start getting serious about making enough product to do something.
So these are not difficult questions.
Thank you.
So
you say.
I don't think they're too difficult.
Look, did I kiss enough stroll ass? I got you, your brother. There's a couple of other brothers I didn't mention. I forgot to mention your mother. Come on.
Next talk. So shadow enhancer a fly thing, or does it you see this kind of thing in other species? You'd think there'd be enough ChIP-seq data in a lot of species to know that. That's the first question.
Yes, I think it's extremely common. The vertebrate types don't like to call them shadow enhancers, and accuse me of seeing something that they've known for years. I mean, there's lots of examples of so-called cis-regulatory redundancy. Somebody finds an enhancer, they knock it out, they don't see an effect in doing sort of a homologous recombination experiment. So anecdotally, at least, I would say it's quite common. It's not going to be, of course, in the case of mammals, it's not going to be temperature, but you can imagine if mom has a poor diet, maybe having a shadow enhancer on some of these genes.
Well, but a
idea.
shadow enhancer is more than just another enhancer somewhere. It's actually an enhancer that's very similar in a regulatory sense than the first enhancer.
Yes.
So you could have a generic enhancer or some. I mean.
Yeah, sure.
So that. Okay.
But I think it's common. I think it's a cheap way to buy you a little insurance. It just makes things a little better. The population geneticists always tell me if it just confers a tiny, tiny, tiny advantage to the population, it will sweep through the population very quickly, certainly in large population sizes like flies.
Okay. And the second question on the second part of your talk is, there's obviously a very nice correlation of the timing with the pause polymerase,
Yes.
but if you muck up the pause polymerase mutationally by NELF or
Yes,
something,
yes.
do you
Yes, we do.
all the usual things that you think you're supposed to do?
Thank God. So we won't exclude you from the reviewers list because we did that experiment. So basically,
I'm
if
the
you...
editor, I'm the editor.
I know, you'll find other reasons. I do understand it. Believe me, I understand that. So I really do understand it. So tail up, shows that steep pattern of activation. If we cross that back transgene into an embryo with diminished levels of NELF-E, SPIT5, or trithorax-like, the GAGA factor, you absolutely get delayed activation. And, again, I think, the way we're thinking about it is more the way people originally thought about enhancers as a probabilistic thing. So if you think about a field of cells, in every given cell, there's a probability of having either a position nucleosome or an enhancer, or a pause polymerase. If it's a strongly stably bound polymerase, then in that field of cells, on average, they have pause polymerase. When the enhancer loops in, everybody's on. If you have a position nucleosome there, it's a stochastic thing because the enhancer has to loop over and over again to finally recruit the polymerase, over and over again to finally kick out that nucleosome and get activation. That's going to seem salt and pepper because that's going to be stochastic from cell to cell. So we really look at it as a cell-by-cell probability of whether you have a pause polymerase or a nucleosome at the promoter, which dictates how fast you get expression. Because again, we're defining activation as 80% of the cells showing nascent transcripts. We're not doing it based on levels, steady state levels of mRNA or anything like that. It's the fraction of the cells that show expression.
Last question.
Mike, right in front.
Yeah.
Regarding shadow enhancers one last time, it appears from your description there is no conceptual distinction between a cis-regulatory element that's kind of near the start site and which happens to be farther away.
Yeah.
In your initial science paper, you adduced evidence that suggested perhaps the faraway guys were evolving faster, that they were somehow different, but I think of it as you can have cis-regulatory elements that are required for a certain level of robustness. Those binding sites might be all glommed together in a kind of long stretch.
Yep.
They might show up in nearby genes. So really there does not seem to be a compelling reason to give CRMs that are a certain distance away.
Don't call them CRMs. Don't call them CRMs. That's such an inelegant term. I can't stand that term.
We can't use that debating technique here. Let's just call it blocks of transcription factors. There doesn't seem to be a sound logical basis to say,
Sure.
once it gets 5 kb away, we're calling it a shadow enhancer. These are proteins that are interacting with the promoter like you've shown, and whether they're all together or split up, does it matter?
My own former postdoc is doing a Brutus on me right here in public. Now,
I told you I'd play the straight man.
Shadow seems to carry so much baggage. I just meant that the enhancer's in the shadow of the gene that it's regulating. That's all. I didn't mean anything friggin' fancy.
5 kb.
I'm kind of a blue-collar guy. I don't know.
KB.
But I agree with you that it is about robustness, and there's many ways to achieve robustness. You could do it with 2 enhancers, maybe better called proximal distal rather than primary and shadow. That's fine. Or you can do it with a mega enhancer that has so-called redundant binding sites in it, as which Kreitman recently showed for EAP stripe 2. You know, what you helped define as the minimal Eve-stripe 2 enhancer is not as robust as the so-called full-length Eve-stripe 2 enhancer that has those extra binding sites next to the minimal 480. So that's another way of doing it. Big, honking enhancer with all sorts of extra binding sites. That'll give you robustness. That'll give you faithful expression when you start playing with temperature and genetic background, or having two kind of minimal enhancers separated. I don't know when you're better off with one or the other. That I don't know. But I certainly agree with you about the nomenclature.
All right, so let's thank Mike again.